Welcome back. In the next few videos, what we're going to do is talk about one of the most confusing and difficult topics in biochemistry, and that is liver metabolism. Now, liver metabolism can be divided into two primary states. One of those is fasted state, and that's what we're going to talk about in this video. The other state is the fed state, and arguably that one is more complicated. The reason it's more complicated is because we can divide the fed state into multiple cases. For example, what if your meal that you just ate was purely carbohydrates, right? Let's say I was to go, and I would never recommend doing this, but go and down a gallon of Kool-Aid or Coca-Cola or something like that. Well, then all you've ingested is carbohydrate, but that's going to be functionally different than a meal that consisted of nothing but steak, right? A steak contains lipids and protein. Arguably, the metabolism of lipid and protein purely is going to be different than a pure carbohydrate meal. So we're going to have to deal with those a little bit differently. But fasted state is for the most part constant. Okay, and we're going to hopefully elucidate a lot of difficult things in this video and make them a little bit simpler. Okay, so we're going to make several assumptions in this video. Number one, we're going to make the assumption that what's happening in our blood system is that our levels of blood glucose are starting to drop. And so we want to maintain our blood glucose within reasonable healthy levels, and that is a narrow range that we want to maintain it within. But it's starting to drop a little bit. So we want the liver to counteract that using negative feedback. We're going to explore how we do that here. We're also going to make the assumption that the primary pancreatic hormone present in the blood is glucagon. Recall from your physiology that glucagon is a peptide hormone that is released from the alpha cells of the pancreatic islets of Langerhans. Okay? And glucagon is going to signal to the liver that we need to run on gluconeogenesis and beta oxidation. Glucagon is going to signal to the liver that we need to run on beta oxidation and perform glycogenolysis and gluconeogenesis. Let's talk about how that is accomplished. Now, if you'll note this picture that's on the left side of your screen, you'll notice that step 1 and 2 are only talking about proteins and amino acids. We will come back to that later. I feel that the best thing to talk about is going to start with the fat metabolism. Okay? Now, what we have to keep in mind is that during the fed state of the liver is going to be happily running on glucose, and therefore it's going to be running glycolysis and the TCA cycle to some extent. Okay? But as blood glucose levels start to drop, there's going to be a decreased reliance on glucose because there's not as much of it, right? And so we're going to have to shift our reliance on energy towards amino acid oxidation and beta oxidation, okay? And ultimately what we're going to find is those two methods of oxidation are going to ultimately be to form glucose and ketone bodies. The acetyl-CoA that we generate through beta oxidation will ultimately be used to form ketone bodies, and the amino acid oxidation of non-essential amino acids will be used to form glucose. Let's explain how that is right now. Okay, the first thing we have to understand is that glucagon is the most prevalent pancreatic hormone right now. So this hormone that we see right here, labeled hormone, binding to its receptor, let's make the assumption that that's glucagon. And glucagon's receptor is a G-protein coupled receptor. So when glucagon binds to its G-protein coupled receptor, and assuming the alpha subunit of the G-protein has picked up a GTP through the action of guanosine nucleotide exchange factor, the alpha subunit with bound GTP will dissociate and activate adenylate cyclase, or sometimes referred to as adenylyl cyclase. Adenylate cyclase catalyzes the irreversible conversion of adenosine triphosphate to cyclic AMP, which we note right here. Cyclic AMP is going to be a molecule that's going to activate protein kinase A through a mechanism that we won't discuss now. Now, this action is done on adipocytes. So you notice here that this cell on which this is occurring is an adipocyte. And in the adipocyte, we have large storages of triacylglycerol. That's what this lipid droplet is right here. In the lipid droplet, there are large quantities of triacylglycerol. And certainly, if you need more help with this part of the video, certainly go review the lipid oxidation playlist. But suffice it to say, cyclic AMP is going to activate protein kinase A. Protein kinase A has two primary targets. Number one, it has a target called a paralipin. Paralipins are these proteins that exist on the surface of the lipid droplet. And normally at rest, in the inactivated state, the paralipins have a protein that's attached. And that protein that's attached is called CG1. Okay? While bound to the paralipin, CG1 remains inactivated. Okay? The second target of protein kinase A is this enzyme called hormone-sensitive lipase. This is a simple serine hydrolase that we'll see the mechanism of in just a minute. Okay? When the paralipin is phosphorylated, CG1 dissociates from it and activates an enzyme called adipocyte triacylglycerol lipase. Adipocyte triacylglycerol lipase catalyzes the irreversible hydrolysis of one of the fatty acids off of triacylglycerols to make a diacylglycerol. In other words, the reaction of adipocyte triacylglycerol lipase is one triacylglycerol to one diacylglycerol and one free fatty acid. Okay, we'll look at the fate of the fatty acids in just a moment. Now we have a diacylglycerol. The newly activated hormone-sensitive lipase, in other words, the hormone-sensitive lipase with the phosphate, can now act on diacylglycerol, and it irreversibly converts the diacylglycerol into a monoacylglycerol and a free fatty acid. In other words, hormone-sensitive lipase in its activated form hydrolyzes off a fatty acid from the diacylglycerol, generating monoacylglycerol. And the terminal lipase is going to be monoacylglycerol lipase. This is going to catalyze the irreversible hydrolysis of a fatty acid off of monoacylglycerol. This is going to generate glycerol, which will ultimately leave the adipocyte and enter the bloodstream, as will the three fatty acids that we generated from complete hydrolysis of the triacylglycerol. And for this video, what we're going to assume is that the triacylglycerol had three saturated fatty acids. Okay? Of course, we can deal with unsaturated fatty acids, but for simplicity, it's easier to conceptualize with saturated fatty acids. Okay, and those saturated fatty acids and the glycerol will enter the bloodstream and go to the liver. The glycerol will ultimately be shunted into gluconeogenesis, and the fatty acids will be run into beta oxidation. But to understand this, we need to understand one thing prior to that. Okay, so at this point, what we're assuming is that the fatty acid has entered the liver cell. Okay, it's traveled through the bloodstream, bound to serum albumin, and when it gets to the liver cell, it gets transported through the membrane, and now it's in the cytosol. Well, now we have this enzyme called fatty acyl-CoA synthetase, which is going to effectively ligate the fatty acid onto coenzyme A. And so what we should get at the end of this reaction is something referred to as a fatty acyl-CoA. Okay, like I said, this is simply a fatty acid ligated to a coenzyme A in a thioester bond. And we note that thioester bond right here, which is characteristic of a fatty acyl-CoA. The fatty acyl-CoA can then go into beta oxidation, and just bear in mind that during times of fasting, the liver cells are deriving most of their energy from beta oxidation. Let's explore how they do that right now. Essentially, what's going to happen in beta oxidation is the fatty acyl-CoA derivative is going to go through several rounds of beta oxidation. In each round of beta oxidation, what you're going to generate is one totally reduced coenzyme Q. This is generated through the action of fatty acyl-CoA dehydrogenase. Okay? Also, what you're going to generate is one NADH per cycle. This is done by the action of beta-hydroxyacyl-CoA dehydrogenase. And also what you should get is what we see right here, and that is one acetyl-CoA per round of beta oxidation, with the exception of the last round, which yields 2. Okay? If you need help with beta oxidation, certainly go view the playlist on that. But suffice it to say, until we get to 4 carbon fatty acids, what we're going to generate per round is 1 acetyl-CoA, 1 reduced CoQ, and 1 NADH. Okay? The coenzyme Q in the reduced state, or ubiquinol, is going to go to complex 3 of the respiratory chain, also referred to as cytochrome C ubiquinol oxidoreductase, and catalyze the pumping of 4 protons into the inner membrane space of the mitochondria. NADH will power NADH ubiquinone oxidoreductase, or complex 1 of the mitochondrial respiratory chain, for the pumping of 4 protons from the matrix into the inner membrane space. This pumping of protons gives ATP synthase power, and it powers the synthesis of ATP by that enzyme. Therefore, the way that the liver hepatocyte gets most of its energy is through beta oxidation by generating reduced CoQ and NADH. Those, of course, power the pumping of protons, which allow ATP synthase to synthesize ATP, and that's the ATP that the cell uses. We will now take a break from talking about fatty acid oxidation and look at the other method of oxidation that is done in liver cells during the fasted state, and that is amino acid oxidation. Now, what is important to understand is this point that I'm about to say, and I want you to listen very carefully. The primary amino acids that get oxidized are your non-essential amino acids. Non-essential amino acids are amino acids that we can biosynthesize ourselves. Essential amino acids we cannot biosynthesize. In other words, we do not have the biosynthetic enzymatic pathways to make them ourselves. We must obtain them from the diet. So the primary amino acids that we degrade during this time of the fasted state are going to be our non-essential amino acids. So what the liver cell is going to do is it's going to take non-essential proteins and hydrolyze them down to amino acids. And we're going to take those non-essential amino acids and run them into non-essential amino acid catabolic pathways, also referred to as amino acid oxidation. That's what the majority of this playlist is going to be dedicated to, amino acid oxidation. Okay? What may not be apparent to you yet is that amino acid oxidation is going to yield 2 primary things. Number 1, it's going to yield ammonia, and the ammonia can be seen right here. Now, ammonia is going to be toxic in large amounts. Therefore, the ammonia will be activated by carbamyl phosphate synthetase 1 into carbamyl phosphate. Then that carbamyl group will be transferred into the urea cycle through the enzyme ornithine transcarbamylase to make citrulline. Ultimately, the ammonia will be excreted in the form of urea. But the other part of the amino acid, which is the carbon skeleton, is going to be shunted into the TCA cycle to be shunted into gluconeogenesis. Okay. Now, if you notice here on this picture on the left, these are all the amino acids and what they get degraded to. And I want you to notice the ones that are in blue. Those are the ones that start here up on the top with leucine and go all the way down to tryptophan. And if you look at all of those amino acids, you should notice that they're all essential amino acids. They are amino acids to which we do not have the biosynthetic pathways to make. And notice how they all get degraded into acetyl-CoA. What this means is that since we're not generally degrading essential amino acids during this time, we're not going to be generating acetyl-CoA through this means. Okay? The amino acids that we degrade are ultimately going to be ones that get degraded into TCA cycle intermediates and pyruvate. Okay, we know that pyruvate is not technically a TCA cycle intermediate, but some of them do get degraded to pyruvate, such as glycine and serine. Okay, these products, which are pyruvate and TCA cycle intermediates, are termed glucogenic products. And the reason they're termed glucogenic products is because they can be put into the TCA cycle to form oxaloacetate, which can enter gluconeogenesis to form glucose. Okay? Let us first discuss the fate of pyruvate. Now, pyruvate during this time is going to go into gluconeogenesis, but something should immediately pop into your head, and that is this. Why wouldn't the pyruvate react with the pyruvate dehydrogenase complex? Well, there are several reasons for this. Let's go into them now. Number one, pyruvate dehydrogenase complex is kept allosterically inhibited. How does this occur? Well, keep in mind that we talked about how the liver cell is doing beta oxidation, and the end products of each round of beta oxidation, with the exception of the last one, are one acetyl-CoA molecule. The last one yields two of them, assuming you start with an even number carbon fatty acid. So you should be generating a lot of acetyl-CoA. That acetyl-CoA will allosterically inhibit the pyruvate dehydrogenase complex. Okay, so that's one way the pyruvate will not get consumed by this enzyme. But if that wasn't enough, the pyruvate dehydrogenase complex can be covalently inactivated as well. Okay, what we have to understand about the pyruvate dehydrogenase complex is it's regulated by two enzymes. Those are pyruvate dehydrogenase kinase and pyruvate dehydrogenase phosphatase. Okay, and if you remember your physiology, kinases attach phosphates to molecules, whereas phosphatases remove them through hydrolytic mechanisms. Okay, now pyruvate dehydrogenase kinase is an ATP-dependent reaction in which the phosphate is transferred onto enzyme 1 of pyruvate dehydrogenase complex. Recall that enzyme 1 is simply termed pyruvate dehydrogenase. When pyruvate dehydrogenase is phosphorylated by this enzyme, it becomes inactivated. So the beauty of this is that when glucagon binds to the receptor, it ultimately activates pyruvate dehydrogenase kinase. And by activating that enzyme, you ensure that pyruvate dehydrogenase becomes phosphorylated. Also, when glucagon binds to the receptor, it inactivates pyruvate dehydrogenase phosphatase. You ensure that that phosphate stays on there and keeps pyruvate dehydrogenase complex inactivated. So that is the fate of pyruvate, and that's why it does not get consumed by the pyruvate dehydrogenase complex and form acetyl-CoA. Now we must talk about the glucogenic products. Okay, those are things that get shunted into the TCA cycle, which ultimately converge at this molecule right here, which is oxaloacetate. Okay, now ultimately, things like alpha-ketoglutarate, succinyl-CoA, fumarate, and all those things are TCA cycle intermediates. And through the action of the TCA cycle, they're ultimately going to form oxaloacetate. And we know from our study of the TCA cycle and gluconeogenesis that there are no oxaloacetate transporters in the mitochondrial membrane. So we have to have alternate means to get oxaloacetate out. The one we'll discuss here is by the action of phosphoenol pyruvate carboxykinase. And the good thing about using this enzyme is it's number one going to convert oxaloacetate into phosphoenol pyruvate, but it just so happens that glucagon upregulates the synthesis of this enzyme. In other words, when glucagon binds to the receptor on the liver cell, the synthesis of phosphoenol pyruvate carboxykinase is increased. Okay, so oxaloacetate will get turned into phosphoenol pyruvate. Now, this is where something should go off in your mind and you should say, hey, why doesn't this just run in the direction of glycolysis? Well, what we're going to talk about in this reaction scheme is we're going to talk about the two allosteric enzymes up through glucose 6-phosphate. Those are phosphofructokinase 1 and pyruvate kinase. Okay. Pyruvate kinase's regulation is simple with respect to phosphofructokinase 1. When glucagon binds to its receptor, it activates adenylate cyclase, which increases the concentration of cyclic AMP. That activates protein kinase A, and one of the targets of protein kinase A is going to be pyruvate kinase. In other words, when pyruvate kinase gets phosphorylated by protein kinase A, it becomes inactivated. Therefore, when glucagon binds to its receptor through a protein kinase A-dependent mechanism, it inactivates pyruvate kinase. That is the mechanism of pyruvate kinase inactivation. But the inactivation of phosphofructokinase 1 is a little bit more complicated. Recall that phosphofructokinase 1 is allosterically stimulated by a molecule known as fructose 2,6-bisphosphate. Now, fructose 2,6-bisphosphate is synthesized by an enzyme called PFK2. Note that that is not PFK1. There are two of them. PFK2 synthesizes fructose 2,6-bisphosphate. However, PFK2 is a bifunctional enzyme. PFK2 is in the same protein as fructose 2,6-bisphosphatase. So what we can say is that, depending on the conditions, this enzyme complex can either create fructose 2,6-bisphosphate through the action of PFK2, or it can destroy fructose 2,6-bisphosphate by hydrolyzing it, and that's by the action of fructose 2,6-bisphosphatase. Well, when glucagon binds to its receptor, it activates protein kinase A. And this bifunctional enzyme gets phosphorylated by protein kinase A. In the phosphorylated state, the part of the enzyme that becomes inactivated is PFK2, meaning that you can no longer biosynthesize fructose 2,6-bisphosphate. But if that wasn't enough, also, fructose 2,6-bisphosphatase becomes activated by phosphorylation by protein kinase A. Therefore, the activity of this enzyme complex is to hydrolyze off the 2-position phosphate from fructose 2,6-bisphosphate to make fructose 6-phosphate. What you ultimately do by phosphorylating this bifunctional enzyme is you terminate fructose 2,6-bisphosphate activity. But remember that PFK1 is strongly allosterically stimulated by fructose 2,6-bisphosphate. But if you eliminate fructose 2,6-bisphosphate, you eliminate the activity of phosphofructokinase 1. And remember that fructose 2,6-bisphosphate is almost absolutely required for the activity of PFK1. If you don't have this molecule, PFK1 essentially turns off. So if you don't have PFK1, that particular part of the scheme you can run in the gluconeogenic direction. And the enzyme that runs in that direction in this area is called fructose 1,6-bisphosphatase. Okay? That enzyme irreversibly converts fructose 1,6-bisphosphate into fructose 6-phosphate. If PFK1 was activated, you'd just reconvert fructose 6-phosphate back to fructose 1,6-bisphosphate. But since we inactivated PFK1, fructose 1,6-bisphosphate will be converted back to fructose 6-phosphate and won't be converted in the other direction. Okay? And also keep in mind that we've inactivated pyruvate kinase through phosphorylation by pKa. So we effectively can now convert phosphoenolpyruvate into glucose 6-phosphate. Okay? Now bear in mind that those glucogenic products that we produced through amino acid oxidation get converted to oxaloacetate in the TCA cycle. They can be turned into oxaloacetate and then be converted into phosphoenolpyruvate by mitochondrial phosphoenolpyruvate carboxykinase. Then the phosphoenolpyruvate can be moved from the mitochondrial matrix into the cytosol where gluconeogenesis can happen. And since these two allosteric enzymes are inhibited, we can now run in the gluconeogenic direction. And basically, by Le Chatelier's principle, if we load our system up with PEP, we'll force all of the reactions to go toward glucose 6-phosphate. The last reaction is going to be by glucose 6-phosphatase, which cleaves off the phosphate through a hydrolytic mechanism, and glucose gets dumped into the bloodstream. Now there should be another burning question in your mind, and the question is this: why doesn't the acetyl-CoA just go into the TCA cycle? Well, the reason has to do with oxaloacetate. Think of it like this. The oxaloacetate that we're generating through the TCA cycle through glucogenic products is being constantly shunted into phosphoenolpyruvate synthesis. And ultimately, that carbon skeleton is leaving the mitochondrial matrix to enter the cytosol for gluconeogenesis. In other words, we're taking oxaloacetate out of the system. We're creating it and then removing it. So this enzyme that would normally be positioned right here, this is citrate synthase. Citrate synthase has two substrates, acetyl-CoA and oxaloacetate. But like any enzyme, it has to have both of them to function. So even though we have acetyl-CoA through beta oxidation, because we won't have as much oxaloacetate, we won't have as much activity of citrate synthase. Okay, so By shunting oxaloacetate into gluconeogenesis, we lose it from the system and therefore can't run citrate synthase. But the acetyl-CoA constantly builds up and accumulates, so it has another fate, and that fate is to run into ketone body biosynthesis, and we will now look at that. Okay, the acetyl-CoA that gets generated is going to react with an enzyme called thiolase. However, in this direction, ordinarily the way you'll hear this enzyme referred to is acetyl-CoA C acetyltransferase. And what we end up getting is this molecule right here, which is acetoacetyl-CoA. Acetoacetyl-CoA is going to react with HMG-CoA synthase, and that's going to generate beta-hydroxy-beta-methyl-glutaryl-CoA. HMG-CoA is then going to react with HMG-CoA lyase to give acetyl-CoA and acetoacetate. The acetyl-CoA that we generate here in HMG-CoA lyase can then go back up here to fuel another synthesis of acetoacetyl-CoA. So we help regenerate the cycle. Okay? But the acetoacetate that we generate here through HMG-CoA lyase is our first ketone body. The acetoacetate can react with two enzymes. One of them is called acetoacetate decarboxylase, which is going to remove carbon dioxide here and give us acetone. We won't deal too much with acetone here, but suffice it to say acetone is one of the ketone bodies that extrahepatic tissues can metabolize for fuel. Acetoacetate can also react with beta-hydroxybutyrate dehydrogenase. Sometimes you may hear this enzyme referred to as acetoacetate dehydrogenase. But in any case, we're going to use an NADH, and we're going to reduce this ketone group right here into a hydroxyl group, which is characteristic of beta-hydroxybutyrate. And so now we have our three ketone bodies. And our three ketone bodies are acetoacetate, beta-hydroxybutyrate, and acetone. These ketone bodies can leave the hepatocyte, they can leave the liver cell, enter the bloodstream, and go to extrahepatic tissues to fuel them. So what have we seen up to this point? Well, we've seen that the major goal of the liver during the fasted state is to provide glucose to extrahepatic tissues and to provide ketone bodies to extrahepatic tissues. Extrahepatic tissues can utilize glucose and ketone bodies for energy, and in other videos we'll actually look at that. So let's do a very, very quick recap. Okay, so what's happening is glucagon is being released by the alpha cells of the pancreatic islets of Langerhans. Okay, that's in response to low blood glucose levels. The glucagon can bind to adipocytes and ultimately cause the release of free fatty acids into the blood. Those fatty acids can then go to the liver, be uptaken, and be oxidized using beta oxidation down to acetyl-CoA. The reduced ubiquinol and NADH that we generate through beta oxidation ultimately fuels the respiratory chain of the hepatocyte and powers the synthesis of ATP through a proton gradient. That powers ATP synthase. The ATP that we generate through that is what powers the hepatocyte and its metabolic processes. Okay, the acetyl-CoA accumulates, and we'll come back to that in a minute. Also, bear in mind that we're also degrading protein here. The rate of protein degradation during the fasting state exceeds the rate of protein synthesis. The amino acids that we generate through protein degradation get shunted into amino acid oxidation, and bear in mind that we're generally only oxidizing our non-essential amino acids, and they generally degrade into glucogenic products. We also generate ammonia, which gets shunted into the urea cycle through the enzyme carbamyl phosphate synthetase 1, generating activated carbamyl phosphate. The carbamyl group gets transferred to ornithine through ornithine transcarbamylase, and we excrete the ammonia in the form of urea through the kidneys. The carbon skeletons of those non-essential amino acids are glucogenic products and get shunted into the TCA cycle, which is shown right here. They ultimately converge at the production of oxaloacetate, and through different mechanisms we can get oxaloacetate or phosphoenol pyruvate into the cytosol. Recall that oxaloacetate can also react with mitochondrial malate dehydrogenase to give us malate, and then we can shunt the malate across the mitochondrial membrane into the cytosol. Then cytosolic malate dehydrogenase can convert the malate back to oxaloacetate, and we can run gluconeogenesis. And that's a handy thing because glucagon binding to the receptor stimulates PEP carboxykinase both in the mitochondria and in the cytosol. Or we can take the oxaloacetate and react it with mitochondrial phosphoenol pyruvate carboxykinase. That'll give us PEP, and we can transport that across the membrane into the cytosol and run through gluconeogenesis. And just bear in mind that PFK1 and pyruvate kinase are allosterically inhibited. Okay. And that's going to give us glucose 6-phosphate. Glucose 6-phosphatase will hydrolyze off the phosphate on the 6th position to give us glucose, which will be exported out of the liver cell into the blood and will go to feed extrahepatic tissues. Now also bear in mind that the oxaloacetate that we generate is being drawn away from the TCA cycle and away from citrate synthase. Citrate synthase no longer has one of its substrates, it only has the one, acetyl-CoA. But without both of them, it's useless. So the acetyl-CoA accumulates and only has another source. The acetyl-CoA accumulates and has only one other path that it can go to under fasting conditions, and that's to ketone body biosynthesis. The ketone body enzymes ultimately convert the acetyl-CoA into acetoacetate, beta-hydroxybutyrate, and acetone, which get dumped into the blood and go to extrahepatic tissues to fuel them. Okay, so I hope this video gave you a little bit of intuition on what's happening during the fasted state. In other videos, we'll actually look at what happens during the fed state under various conditions. See you in